My name is Liuji Wu. I'll introduce the paper submitted to iMeta. The title is a comprehensive atlas of endogenous peptides in maize. Endogenous peptides are increasingly recognized as vital regulators of plant growth, development, and stress responses. However, majority of the research on endogenous peptides in plants has so far been limited to individual peptides or small groups of peptides, often focusing on their roles in specific biological processes. While transcriptomics and proteomics have advanced our understanding of gene and protein regulation, peptidomics, the large-scale study of endogenous peptides, have not been comprehensively analyzed on a genome-wide scale in plants. In this study, we present a comprehensive peptidomic atlas of 13 maize tissues. Using a three-frame translation of maize canonical coding sequences, we identified 6,100 nonredundant endogenous peptides, significantly expanding the known plant peptide repertoire. These peptides were then subjected to analyze their abundance pattern, genome-wide distribution, and their co-expression with the source and neighboring proteins at genomic scale. These peptides presented variation in abundance pattern throughout maize development. The tissue-enhanced and mixed peptides demonstrated more dynamic expression across both vegetative and reproductive phases compared to tissue-specific peptides. This highlights the complexity and dynamic nature of peptidome. We found extremely low correlation between most peptides and their source proteins. The source proteins exhibiting higher expression correlation with the endogenous peptides were mainly enriched in cellular metabolism, including the primordial glycolysis pathway. The WGCNAR package was used to group similarly expressed peptides, or peptide source proteins across maize development into modules. The peptide module zygon genes exhibited different abundance patterns compared to the protein module's eigen genes throughout maize development. This suggests that peptides may perform distinct biological functions from their source proteins. Furthermore, the peptidome may operate through distinct regulatory mechanisms to have specialized roles in developmental processes. We found extremely low correlation between most peptides and their source proteins. The source proteins exhibiting higher expression correlation with the endogenous peptides were mainly enriched in cellular metabolism, including the primordial glycolysis pathway. In summary, we generated a highly reliable peptide atlas of maize, offering novel insights into the dynamic expression patterns and tissue-specific distribution of endogenous peptides across developmental stages. By integrating peptidomic co-expression profiles with previously reported proteomic profiles, we found that the peptide abundance did not consistently correlate with the abundance of their source proteins, suggesting the presence of complex regulatory mechanisms. This comprehensive atlas can be integrated with other omics data, such as transcriptomics and proteomics, to enable a more holistic understanding of gene regulation and function in plants.